Good morning, everyone, and welcome to our Whiteboard to Boardroom Technology Snapshot webinar series. My name is Jim Baxendale. I work with the Innovation Center Whiteboard to Boardroom program. Some of you that may be new to our webinar series, these technologies from our institutional partners are available for licensing in many cases as new startup companies or new product opportunities for established companies. We also like to take this opportunity to receive input from some of you in attendance on commercial opportunities or other questions you may have about the technologies. Feel free to use the chat feature for questions at the end of the presentation, or you can ask them in person if you like. We do record these so that the link to the recorded presentations will be sent to you following the webinar. Feel free to share the information about these webinars with your colleagues and others that might have an interest. We do like to get this out to your networks as well, and we'd be happy to visit with anyone there that may have interest. We have two technologies today, both from the University of Missouri St. Louis. Before I quickly get to that, I just want to put in a plug for the next webinar on September 11th, which will have a presentation by physicians from the University of Kansas Medical Center, one on a kind of a novel, portable, single patient negative pressure pandemic tent. The second one is from the University of Missouri St. Louis, again, around cybersecurity. So look forward to having some of you in attendance for that. So first off is Professor Gogol from the University of Missouri St. Louis. And with that, if you'd like to start, that would be great, Dr. Gogol. Thank you.
Thank you. If we could have the first slide. Pneumonia is a pathology in which the inflamed lungs fill with fluid as a result of bacterial or viral infection. Hospital-acquired pneumonia, called HAP, and ventilator-acquired pneumonia, called VAP, are caused by resistant bacterial infections acquired during hospitalization. HAP-VAP affects about 375,000 people in the US each year, and it has a mortality of between 30 and 50%. Treatment can take as long as 25 days and cost the hospital up to $64,000 per patient. Comorbidities and increased antibiotic resistance make current treatments ineffective. Dr. Daniel Hoft, a physician at St. Louis University Hospital, told us that, quote, HAP-VAP is the biggest problem among already sick patients because it has the highest medical complexity. An effective treatment for HAP is clearly required. Could I have the next slide, please? The CDC proclaimed in 2019 that we already live in a post-antibiotic world owing to bacterial resistance. MRSA infections have diminished, but the MDR Enterobacter infections that cause HAP-VAP have dramatically increased. The dwindling antibiotic pipeline helps define this challenge. Of 42 new antibiotics in development, no new antibiotic class in development in the US appears to address the problem. A new antibiotic for HAP is urgently needed. Next. Oh, sorry. This is the slide?
No, this, no. Yeah.
We need the...
Yeah, this is good.
Okay. We've developed a non-intravenous antibiotic as a first-line treatment for HAP. Our combination antibiotic consists of a novel adjuvant that we call Synamp, and will be paired with existing antibiotics in a single vial as shown in the image in the corner. Synamp does not kill the bacteria by itself, but overcomes resistance to recover and enhance antibiotic potency. The bacteria tested did not develop resistance to Synamp in 30 days. Although at the preclinical stage, we've confirmed 64-fold enhancement in norfloxacin potency against multi-drug resistant gram-negative bacteria. We've demonstrated that toxicity and drug interaction occur only at greater than 50-fold of IC50. Our drug is bioavailable in mice at 200 nanomolar from a dose of 0.5 milligram per kilogram in a single trial, and their half-life is 1.5 hours. We're working to identify a lead combination with a once-a-day IV therapy in conjunction with a broad-spectrum antimicrobial. Next slide, please. Our SynAmp adjuvant is a new structural class. It is a non-toxic amphiphile that selectively inserts into bacterial membranes due to the presence of 6 basic nitrogens. It increases the antibiotic's permeability into bacterial membranes, but not of human membranes. It disrupts the resistance mechanisms dependent on ion gradients, so drugs are not pumped out. The adjuvant can be used with a range of FDA-approved antimicrobials, making it a new platform. At present, 1 patent has issued, claims are being negotiated with the PTO in a second, and a quick but limited allowance has been offered on a third. The patents are owned by UMSL, but we'll license the technology after completion of a certain stage of funding and de-risking the technology. Next slide, please. Our competition can be divided into generics such as Zosyn, Miropenem. Sorry, we're at 6. Oh, I'm sorry. We missed a slide here. Well, that's okay. The antibiotics to treat a HAP patient cost between $2,500 and $5,500. 375,000 US patients contract HAP annually, making the total US market approximately $2 billion. The payers we interviewed were amenable to paying a premium for a more convenient product, roughly in the range of $5,000 to $12,000 per HAP patient. Our product will be combined with several antibiotics to address multiple different antibiotic resistance problems. An alternative market segment recommended by several of the physicians and easily accessible by acquiring parallel indications would be complex urinary tract infections, CUTI or UTI, which need better interventions. Next slide, please. Competition. Where? Oh, we got them out of order, I'm sorry. Our competition can be divided into generics such as Zosyn, Miropenem.
Good morning. Pretty good.
And newer branded drugs.
Oh, yeah.
I'm sorry, what's going on?
We got him muted. Go ahead.
Also,
our competition. Our competition can be divided into generics such as Zosyn, Miropenem, and newer branded drugs such as Zerbaxa and Avicaz. These antibiotics comprise a known drug class combined with a beta-lactamase inhibitor. SynAmp is a new chemical entity with a new mechanism of action that would allow physicians to adopt without fear of further resistance spread. Ours would be the first non-resident antibiotic of its class, leading to extended clinical use. Our pricing will be higher than generics, but competitive to branded drugs to increase adoption. Next slide, please. Okay, we've been through that. Next slide, please. Regulatory strategies. The clinical studies and the cure rate will be the focus of our new class of adjuvant. Since HAP is an unmet need, we're eligible for multiple expedited pathways, including, but not limited to, fast-track approval and a priority review. This will reduce the number of clinical trial participants to 300 and the overall funding required to reach market. Will be eligible for an early we're currently at the preclinical stage of development, and reaching this early IND meeting is our goal. We note that the antibiotics have the shortest development time of any drug class. Next slide, please. These are our milestones. To reach the early IND meeting goal, we'll need to complete the following non-GLP in vivo studies. Milestone 1 will identify a lead combination of SYNAMP and antibiotic against appropriate bacterial strains. Every milestone thereafter will serve as a killer or no-go experiment. By the way, all in vivo studies will be conducted by a CRO. Milestone 2 will establish dosage and how much drug reaches the lungs. Milestone 3 will focus on toxicity, and milestone 4 will determine the cure rate in HAP rodent models. High toxicity or side effects or low in vivo efficacy will be no-go signals. After the early FDA meeting, we can conduct GLP and CMC studies. Next slide, please. This is our team. You can see all of our smiling faces here. And I think there might be a last slide that's a summary. And there is. To summarize, the critical unmet need is a new treatment for HAP-VAP that can counter increased bacterial resistance. Our product is a first of its kind, non-resistant adjuvant antibiotic for the treatment of MDR bacteria causing HAP. This is a platform technology applicable to several drugs and organisms. The technology development has been supported by federal funding and by the system. Success to this stage is documented in 5 published articles, 1 issued, and 2 pending patents. The urgent need means we'll have an expedited clinical development pathway that we estimate at 3 to 4 years. Our preclinical scientific and business team is experienced, balanced, motivated, and has great smiles. We're asking the $200,000, this was for, we're not asking you for money. Sorry. That's our talk.
Thank you, Dr. Goker. Questions come up. Is there any data or belief that would suggest that SYNAMP would work with any other compounds beyond antibiotics?
Yes.
You
Or not data.
want to answer that? No. Yes, there is. We have some data, but we don't really want to talk about it.
Well, we don't have data for anything other than antibiotics.
Yeah, we have very good data for antibiotics. We have some preliminary data in other areas that we haven't disclosed and haven't discussed.
Okay.
I apologize, but Tamara is on this meeting and she would kill me.
Okay. In terms of executive, what are you looking at in terms of expertise, background, experience for that individual at this point?
Well, we're mostly scientists. Moed, who's sitting at my side, is an entrepreneur as well as a PhD microbiologist. He was named 30 under 30 by the St. Louis Business Magazine as an entrepreneur. And so he has a lot of business background. But what we really need is someone who has some scientific background. We obviously provide most of that, but we need somebody who knows how to contact the business community, the angel community, the venture capital community, and so on, so that we can get our project rolling better. We also need somebody who has organizational sense, who can tell us that the market is such and such and that we really should go, say, in the UTI direction rather than the HAP-VAP direction, based on whatever business observations there are. So we're looking mostly for business expertise, someone who also has a scientific background who can give us a different perspective would be terrific also.
Okay. You mentioned earlier about the license from UMSL, and I don't want to get into specifics, but is that been negotiated at this point, or is it kind of in negotiation with your startup?
Well, we've talked extensively with Tamara about licensing, and we have not taken a license yet because we have patent obligations that are expensive. We don't have a product yet that's generating revenue, and so we've delayed taking the license until we have somebody on the hook who can help us financially.
You also mentioned seeking drug developers. What does that mean to you?
Well, we're looking for a collaborative partner, ultimately, someone who could help us or who could take over doing the clinical trials. We sort of are working in 3 stages. We're doing the more or less academic side of this, developing compounds and doing non-GLP testing in our lab. Second stage is we have used a CRO, and we're planning to use additional CROs to do animal testing. We think that good animal data will allow us to try to partner with a company that is capable of taking over the really big issues of regulation and clinical trials. Did that answer your question?
Yeah, sure did. Yeah. Yeah. Any other questions out there before we move on?
I do not have any in the chat.
Yeah. Okay.
Okay. Thank you for the opportunity.
Okay, thank you. Yeah.
Let's listen to Mike.
Okay.
Hey, good morning, everybody. I hope you can hear me well. I'd like to thank Jim and Robin for helping me to be involved in this program, and particular thanks to Tamara Wilgers, our technology transfer specialist at the University of Missouri St. Louis, who's just provided a wealth of help and guidance for most of the technologies at UMSL. So my name's Mike Nichols. I've been at the University of Missouri St. Louis for 16 years now. And during that whole time, we've been studying the underlying mechanisms of Alzheimer's disease. In particular, we focus on the protein aggregation component and the neuroinflammation components of Alzheimer's disease. So I think most of us here, in fact, all of us understand the impact of AD, both on a personal side and a financial side. So I'm not going to belabor everybody with too many statistics, but just know that currently, if you have a diagnosis of AD, it's 100% fatal. There's no approved treatments right now that actually modify the disease. So there's some drugs that can treat the symptoms of dementia. So some of you may be familiar with Aricept or Namenda, and they're very effective drugs, and they'll buy a little bit of time to stave off the dementia, but they don't actually attack the disease progress. So what we've done over the last 4 or 5 years is work on an antibody. Next slide, please, Robin. That is attacking part of actually the main trigger for Alzheimer's disease, and that's the amyloid beta protein. So at the core of AD is a protein aggregation phenomenon. And this you see in other neurodegenerative diseases, and for all of them, there's no treatment right now. So the main trigger for AD is this accumulation of A beta protein in an aggregated state. I wanted you to turn your attention to the right side of the screen. And this is a schematic of how A beta, the protein, goes from a single monomer protein to an aggregated state. And it's these aggregated states which actually wreak the havoc in the brains of Alzheimer's patients. And so what you'll typically see in a brain slice on the left side is the A-beta fibril plaques. But most researchers now believe that the intermediate structures, such as protofibrils, which you see on the right, may be the more deleterious protein species. Next slide, please, Robin. So our solution is an antibody that selectively targets these protofibril structures. And so we started early on with some serum antibodies, and we've published that work and done a significant amount of characterization. In the last 2 years, we've developed a monoclonal antibody. And both of these serum and the monoclonal antibodies are very selective for this protofibril form of A-beta. So the idea is to immunize early AD patients with the monoclonal antibody. And this is not a new idea. In fact, antibodies have been the primary target or therapy for AD patients, or at least in clinical trials, over the last 4 or 5 years. Now, I will acknowledge that some of these antibodies have failed in the clinical trials. None of them have succeeded yet. So researchers have turned to selective antibodies that target one species over another. So I showed 2 on the right side of the screen, aducanumab and BAN 2401. 2401 is the most similar antibody to ours. It's currently starting phase 3 clinical trials. Aducanumab has been reintroduced in a phase 3 trial with higher concentrations. But what sets our antibody apart from these 2 antibodies is the selectivity. So if you look at the A-beta oligomers in the middle of that table, and you look at the plaques, the other 2 antibodies will target both of those. Our antibody will target one of them. We feel like this selectivity is going to give us an advantage. I'll talk about what we've done later, but suffice it to say that we've cloned, sequenced, expressed, purified, and characterized the monoclonal antibody in our lab. So I've got, next slide, please, Robin. I want to follow up on this selectivity difference here. So an example in panel A is the 2401. So that's a dot blot assay, and it shows that even at the lower concentrations, the antibody recognizes both protofibrils and fibrils. If you look at our selective antibody, 113, it targets just the protofibrils and doesn't recognize the monomers and the fibrils very well at all. Now, another advantage that we have is that in the process of identifying these monoclonal antibodies that are selective, we found some that are non-selective, that are prepared in exactly the same way. So you see that in panel C, this 513 antibody, which is a rabbit monoclonal IgG, recognizes the protofibrils and the monomers with the same affinity. This is going to allow us to test these 2 antibodies later and see if the selective antibodies make the difference. Next slide, please, Robin. So the idea, which again is not new, is an IV delivery of monoclonal antibody solutions on a monthly basis. We identify early stage AD patients, which is mild cognitive impairment and mild AD, and they would receive the monoclonal antibody solution. Next slide, Robin. So the impact and the commercial potential of an AD therapy, since there is none right now, is significant, to say the least. So we would anticipate a first year sales of 100 million, which is about 10% of the market. And that's if this therapy made it through the complete phases of clinical trials. And then a market of about 1 billion after 5 years. That's based on the market growth and the growth in the number of cases, which is increasing with AD. Next slide, Robin. So this slide can be a little confusing, so I just want to elaborate a little bit. The only approved and prescribed treatments right now are the first two, and they do not stop AD progression or reverse any of the AD effects. They can provide a short-term relief. Everything else on this table has not been approved yet, but there are researchers targeting inflammation, targeting tau, targeting A beta protein. Tau is another protein in AD that causes problems. And so everything on the rest of this slide is all theoretical. And what the researchers and companies believe they will do. So the antibodies that target A beta are believed that will stop AD, and we believe that with our monoclonal antibody. And again, what sets ours apart from the other two is the selectivity of that antibody. Next slide, Robin, please. With Tamra's help and the attorneys at University of Missouri St. Louis, we filed a patent application on these antibodies that include a whole class of antibodies and some of the methods that we've developed. Next slide, Robin. Again, we've cloned the variable regions for both the heavy chain and the light chains for a whole class of antibodies, both selective and non-selective. And then we're able to express and purify these antibodies in our lab. Next slide, Robin. So monoclonal antibody treatment for AD would be a new drug, eligible for fast track review. We anticipate the clinical trials will be about 5 to 7 years for phase 1, phase 2, and phase 3. Right now, there's no approved comparable, although as I mentioned, one of the antibodies in phase 3 right now has some similarities to our antibody. Okay, next slide, Robin. So what do we need to do to get to the clinical trial phase? We think we have some critical experiments that need to be done in transgenic mice. So these are mice are engineered to get Alzheimer's disease. We want to test both the diagnostic ability of this antibody and the therapeutic ability of this antibody. And the fact that we have the selective and the non-selective antibodies available, we can actually prove the concept in these AD mice that the selective antibody would be the best potential treatment for AD. The last step would be to humanize this antibody to reduce any inflammatory reactions in human patients. We've got a team at University of Missouri St. Louis. We've set up a company called Antibody STL. We also have a team at University of North Dakota Medical School, and we're working with the Animal Surgery Corps at Washington University Medical School to do these particular experiments, and most of these would be considered killer experiments for that. So I'll end on that note, and I'll be happy to answer any questions you may have.
I noticed you listed amounts of funding needed for various aspects of your milestones there. do you have grants that can support that work, or are you going to be seeking SBIRs or other funding?
Yeah, so we're writing, great question, Jim, thank you. All of those things. We're looking for private investors, of course, in this. We are getting ready to submit an STTR in conjunction with UMSL to the NIH. And I'm also writing basic research grants that follow up on how this antibody works and how it recognizes the particular conformational epitope on these A beta protein species. So we're looking at all kinds of options for funding in this, Jim.
And you mentioned the antibody in clinical trials that has similarities. Can you
Yes.
expand on that a little bit?
Yeah, BAN 2401, the original papers were, the antibody was called MAB158, and then this group humanized it and renamed it as BAN 2401. And that has significant industrial support and pushing it through the clinical trials. And it looks promising. And so I view that as a good sign that we're on the right track and that we could be a competitor for that. So I actually hope that that antibody succeeds and that our selectivity ends up giving us an advantage even over that antibody itself.
Okay, let's see. And in terms of the infusion process, anything different about how that's being done now?
Yeah, so we're not trying to develop any new ways to deliver the antibody. We're just building on the current methods for administration of monoclonal antibodies, which I should say is not limited to just the Alzheimer's field, being used in a number of other biomedical fields to deliver treatments for that. Arthritis, cancer, other fields are using it. So we're just utilizing their techniques and their strategies. And that clearly would not be done by us. That would be done by physicians, and once the antibody made it through all of the phases of clinical trials, including safety and all of that.
Yeah. So in terms of the attendees we have in the webinar today, we can't obviously get into asking for funding necessarily, but obviously you need certain funding. Is there any other needs that you have for strategic partners, advisors, mentors? It sounds like you've got some good collaborations going on, but is there any other asks that you have or needs that you have?
Yeah, I would say that the business aspect is a big one for us. I think we're in good shape research-wise, and of course we can always use expertise on how to navigate clinical trials, and of course that would be a sizable investment, and we would have to collaborate with a large investor on that sort of thing, and particularly someone that was experienced at taking a drug through that sort of process. But I would say right now, certainly expertise in the business aspect is something we could lean on right now.
Okay, good, very good, okay. Any other questions out there? I don't see anything in the chat right now, but any others?
Hey, Jim. Dennis Radnauer, BioNexus PC.
Yeah.
Hey, Mike, great presentation. Question for you. You talked about a couple things that you're looking to do, one of which was scale up manufacturing. Do you have connections there to be thinking about that already? What's your manufacturing capabilities currently?
Yeah, so we can actually, I mean, we can make a sizable amount of the antibody if you're doing research type assays and, you know, maybe marketing the antibody to researchers that might use it. And we can probably handle the scale up for the animal studies. Now, of course, the scale up for clinical trials is several orders of magnitude probably beyond what we could handle unless we expanded our capabilities in a significant way. I will say just down the street from UMSL is Thermo Fisher, and that is the research arm of Thermo Fisher that used to be called Patheon. And that's what they do, is scale up monoclonal antibodies. And we have a number of students that graduated from UMSL at all levels, PhD, masters, and bachelors, that are working there. So I think we can probably strike up some collaborations with them or utilize their expertise. But it doesn't have to be them. Scale up for us could come from a variety of sources when it gets to that level.
Yeah, that sounds good. I think that was one thing I was going to offer, was just some connections there. I think Catalent Pharma Solutions is here in Kansas City. I think Thermo or Patheon would be good as well. And I think Jim's probably got some good connections through Kelly Gillespie there, higher up as well. Hey, one last question. I might have just missed it.
What was the first one you mentioned before Thermo?
Catalent Pharma Solutions. They're here in Kansas City in the old Marion Labs building, and they do a lot of kind of early stage drug development. I don't know about the biologics approach, if they do kind of monoclonal.
Okay.
But
Yeah.
I could definitely make some connections and help find out if they do.
Great.
The one thing I might have missed, I've got kids running around, so I stepped away for just a second, but you talked about how this targets oligomers rather than the plaques, and that specificity is a really key differentiator. Are there studies, did you mention or describe studies where targeting the oligomers has positive outcomes in animals, or is that the transgenic work that you guys need to do?
Yeah, so great question, and it's really the crux of the overall question of this whole strategy. And nobody has actually tested that hypothesis. They've developed antibodies and tested these antibodies, but not compared the two. And that's what we're hoping to do, is compare the selective antibody with the non-selective antibody to show that it makes a difference. Right now, it's all hypothetical, and no one's actually tested that. Now, people have targeted things in much smaller scale in cell culture models and shown that antibodies, but still not done a real rigorous comparison, but shown that these antibodies can work. But your question hasn't been answered yet. And we'd like to answer that question in a killer experiment.
Yeah, it seems like and it would be a pretty crucial experiment, right?
Yeah.
And everything hinging on that.
It would.
One last note. I have a good contact at Takeda out in Boston, and I send him technology sometimes that are in certain areas of expertise. I feel like this would be one. And so maybe, Jim, you could just help Mike and I connect, maybe offline, and I can try and make an introduction out there.
Okay. Yeah, sure. No problem.
Great.
Thanks.
Yep. Thanks, Dennis.
Okay. If there are no others, or if something comes up later, just drop me a note. But thanks again to both of you. This has been great, and I appreciate the questions from our attendees, and look forward to hearing from you again in September. So thanks again, everyone. Have a great weekend.
You too. Thanks, everyone.